And the next in line, we have another great scientist today here, Nils Gehlenborg. Nils is currently an assistant professor in the Department of Biological Informatics at Harvard Medical School. He's also director of the Masters in Biological Informatics program and director of the Biomedical Informatics and Data Science Research Training program. Correct me, Nils, if I got it wrong. Nils focuses on developing visual interfaces and computational techniques that enable scientists and clinicians to interact with biomedical data quite efficiently. And he's therefore closely collaborating with many clinicians and experimentalists, allowing him to integrate algorithmic approaches from biomedical informatics with advanced data visualization techniques, as we will hopefully see today. So I'm already quite excited to see what Nils has prepared for us in his talk about visualization of spatial single-cell omics data. So Nils, floor is yours.
Thank you, Jan, for the introduction. Thank you to all organizers for the invitation to present here today. It's great to see that there is now a spring school in biomedical data visualization, and I'm very happy that I can be part of this inaugural event. I will indeed talk about visualization of spatial single-cell omics data. And my goal today is to give you an overview. I understand you're all coming to this school with very different backgrounds, very different levels of experience in, I assume, data visualization as well as biology and medicine. So I'll give you sort of an overview of what is exciting in this field. And maybe I can convince a few of you to actually start working in this area. And if you're staying here through the end, well, maybe you'll actually start working with me in this area, because I certainly have some open positions in my lab for people who are interested in molecular biology, single cell biology, and data visualization. So let me get started by giving you a super quick overview of single cell and spatial biology. So to date, people have done biology like they've treated their apples, they've turned essentially most of the things that they've studied into applesauce. So as you know, when you're making applesauce, you take your apples, cut them up into small pieces and cook them and mush them, and in the end you get some fairly homogeneous sauce that might be tasty, but is actually very different from the starting material, right? For applesauce, it's apples. And in the case of biology, clearly we're not starting with apples. We're generally starting with tissues or cells. And depending on sort of when you get into, or at what point you sort of start looking at molecular biology and cell biology, back in the day, and that day is not even over, people would just look at tissues in form of mushed up, ground up tissues, maybe with a few specific cell types extracted, but generally it was just a mix of all the cell types that are in a tissue. Say in your brain, as you know, you have many different types of cells, and that's true for all other organs. That's true for your skin, that's true for your kidney, for your liver, for your heart. They're all composed of different types of cells. And the question that we're trying to answer today is by actually not only just grinding everything up into applesauce, but by looking at the individual apples, or looking at individual cell types in these tissues and understanding the relationships between these different cell types, and in particular, the spatial relationships between these cell types. So that is what we're aiming at in single cell and spatial omics data, is we're trying to understand relationships between different cell types. We're trying to maybe first of all try to establish what cell types are there in a specific organ, say in your brain or in your kidney or in your liver, to essentially come up with the census of cells that are available, and then figure out what all of these different cell types are doing, how they're interacting with each other, and what we can learn about biology when we start moving from applesauce to apples, essentially. And it should not come as a surprise that this is very exciting. And one of the better known journals in sort of biological technology and methodology development, which is Nature Methods, they they have a method of the year every year, and they've had this for probably at least a decade. And so in 2020, the method of the year was spatially resolved transcriptomics. So transcriptomics, for those of you who are maybe a bit newer to biology and molecular biology, the transcriptome is the set of all transcripts, meaning it gives you a sense of which genes are turned on or off in a specific tissue, or in this case, in a specific cell. And that is what a transcriptome can tell you. And spatially resolved transcriptomics essentially is a way to measure in a tissue, in situ generally. So or it can be done in situ, it can also be done in a slightly different way, but you can measure in a tissue, in every single cell, which genes are turned on and turned off, or at which level genes are expressed. Now, this can be done not quite at the scale of the whole genome generally, but fairly close to thousands of genes in every single cell in a tissue. And what we get out of this looks something like this. So this is an example for a visualization of spatially resolved omics data. In this case, what you see here, this is a fairly simple visualization. Actually, there's the spatial view over here, and what you can see in the spatial view are measurements for about 39 molecules. So there are only 39 different genes that were measured in this example, but there are about 2 million instances of the transcripts for these genes that were measured in here. So there are 2 million data points in this visualization. In addition to that, what the authors did in this project, they then mapped the activity of these genes to cells. So they identified individual cells and then got cell level measurements for these 39 genes. And there were about almost 5,000 different cells, which are shown here in these two projection views, which we'll get back to what they can tell us about biology. And so here's the spatial view. Here are scatter plots that represent individual cells. And you'll see in a moment that these cells can also, that these spatial views can be shown at different levels of resolution, either at the individual molecule level or at the cell level, or you could even start combining cells into so-called functional tissue units into sort of a higher level of resolution. The heat map down here shows us for these 39 genes, which are the rows in this case, across those almost 5,000 different cells, what the expression profiles look like for these cells. And the one thing that you can see in here right now, but that is also present, is behind these individual molecules, which were just measured as present or absent. You can see, or you would be able to see, if you were looking at the interactive version of this tool, you can see an image that corresponds to the tissue, to a tissue slice. In this case, it's in the brain. It's a somatosensory cortex in mice, and you would be able to see the actual image that was taken, or an image that was taken along with these measurements that were made. Okay, so the main takeaway for this first part of this talk is that you remember really the difference between apples and applesauce. That's one of the really key differences in biology, how some or many in molecular biology, genetics, genomics are pursuing biology today. So trying to look at individual cells so that we can study relationships between cells and understand composition of tissues and interactions within the tissues, rather than just looking at, say, a kidney as one homogeneous mass of cells. Okay, so what do we do for exploratory analysis, and what do we need to do if we want to visualize this kind of single cell data? So in spatial and single cell data visualization, one of the major challenges is we're dealing with an extremely wide spectrum of data modalities. So what are data modalities? For example, images are a specific type of data modality. But now you can generate images by using optical microscopy and say fluorescence markers to obtain an image. But you could also use imaging mass cytometry or imaging mass spectrometry, or one of the many other assays that are being developed today to generate imaging data. So this is always intrinsically spatial, of course, when you take an image of some tissue. The other type of data modalities, or the group of data modalities, are the omics assays. So these are generally based on some form of sequencing of transcripts. Sometimes they're made visual directly in the tissue. Sometimes cells are separated and then sequenced individually and after they've been sorted into groups. So you can do that too. So those are some of the challenges you will deal with if you're trying to visualize this kind of data. You need to be able to bring visualizations from many different data modalities together. Another challenge that you'll be facing, and a challenge that needs to be addressed for visualization of this kind of data, is that the data exists at many different scales, which is always exciting when you think about visualization, that you can navigate across scales. Of course, you can apply, Schneiderman's information-seeking mantra with overview first, zoom and filter, details on demand. But in this case, you definitely need to apply this because you can get information at the organ level, or parts of organs, or functional tissue units, or even at this individual cell level. Functional tissue units are groups of cells that perform a certain type of function within an organ together. And of course, another thing that you'll deal with is the actual data and where that data is coming from and how people are processing this data. And so you'll see there's a rich ecosystem of analysis tools in R and Python that you somehow need to be able to support if you want to build a tool for real-world use. And why does all of this matter? Where is this interesting, and why should you maybe start thinking about visualizing this kind of data? Certainly, funding agencies such as the National Institutes of Health or the Chan Zuckerberg Initiative have discovered that a lot of future insights into how organisms work will be at this level of single cell and spatial biology. So there are a lot of efforts ongoing to study tissues at that level. And there are three examples here: the NIH Human Biomolecular Atlas program called HubMap, or the NIH Human Tumor Atlas Network. Those are projects funded with tens of millions of dollars to generate data of this type that I just described across different tissues, in case of the Human Tumor Atlas Network, also across different cancer types. The Human Cell Atlas is a sort of more grassroots effort to study single cell data and to essentially establish an atlas of the human body at the single cell level. The Human Cell Atlas has different sort of sub-efforts that are looking also in some cases at spatial data, but they also have a lot of single cell data that is not spatial, where you have no spatial information about the individual cells. As I said, all of these efforts are generating lots of different data modalities. In many cases, these are exploratory projects, right? So these are not projects that have a clear hypothesis. These are projects where the first step is to generate data and then to start gathering insights and generating hypotheses based on that data. And so that is why exploratory data visualization plays a critical role in these efforts. And all of these efforts also have specific funding to support data visualization, which makes this whole effort even more exciting, right? And the last comment here is something that I also want like you to take away from this and to remember, is that when you're dealing with this kind of data, you're dealing with enormously large data sets. They generally cannot be moved around. You not believe it, but even today in the Human Biomolecular Atlas program, we have data types where people need to ship around hard drives because it's just too much data to even transfer it over the internet. So that is a challenge that you will be dealing with in certain cases, that you need to build visualization tools that can operate on remote data sets. So here's just a quick overview of HubMap and their data portal. Which I should add for full disclosure, I'm part of this project, and here's just sort of an overview of the different assay types or data modalities that are available in this data portal today. And this is only 3 years into the project. This project has another 5 years to go, most likely, and there will be many more data types that will be produced and that need to be integrated and visualized. So how do we generally visualize single cell data? I've already given you an overview. And I want to just revisit this for a moment, because it is actually, when you think about it, there's not that much in terms of cutting edge, brand new visualization types. Most of you will be familiar with what I'm going to show you now, but the exciting and the challenging part is much more in how to bring all of this together to gain insight into biology. So good old scatter plots, very, very common in single cell biology, because you can represent every single cell as a data point, apply a HuMap or a t-SNE projection, for example, and you'll get these beautiful maps, which of course, and not for today, but as you probably all are familiar with the challenges of applying, say, t-SNE, choosing the right parameters to get what you want. HuMap a bit simpler with regard to parameters, but still you need to choose these parameters correctly. And then hopefully what you'll see is that you're able to get a map that contains clusters, and then you'll hopefully figure out that these clusters correspond to certain cell types. And what does it mean to correspond to a certain cell type for a single cell? This is generally derived from biomarkers that were measured in these cells. So for example, it could be transcripts in the cells, so that you look at the expression levels of a number of genes that determine what cell type that is. That is one way to use molecular information to determine cell types. For pathologists, they might, in some cases, look at a cell and be able to tell what cell type it is. But here we're really relying on molecular data, omics data, essentially, that was measured in these cells. So you could also, instead of using RNA sequencing data, for example, you can also use multiple fluorescent markers that target different proteins that are expressed in these cells, and then use combinations of fluorescent markers to determine what cell type you might be looking at. So there are many different approaches to determining cell type and cell identity. And that is all, under very active development. There are many, many different approaches that are out there that people are trying to establish and they're trying to improve. But in the end, what it boils down to, as a visualization expert, you would want to know what is a cell type, or what is sort of a suspected cell type, be able to map it to the cells, maybe with some amount of uncertainty, label these things, and then not only have sort of the cell type label, but also be able to display what are the molecular markers that were actually used to make this determination what cell type it is. And that's where heat maps come into play. Here's essentially the same data, and you see the cluster assignments here, or cell type assignments, are shown here on the left in these colors, and then you see that across the genes across these cells have different expression patterns. And this is another way to understand sort of whether your assignments and your clusters make sense. Most importantly, of course, we want to have spatial views. And this is a very simple example of a simple fluorescence microscopy image with 2 channels in this case. So what we're looking at, this is actually the same data set that you saw earlier. So there are cells that were segmented. These outlines were slightly simplified. In this case, they are usually a bit more jagged, but here we just simplified the polygons for computational reasons. And then you see here in this case, poly T is blue, and then nuclei are colored in green. You also see these algorithms are often not very good. So there is a cell that was missed. The nucleus is completely without an outline. But so with that, we get spatial relationships, right? And so now for each of these cells that were identified here, we generally have this vector of markers that, for example, is expressed in this heat map, that we can use to not only look at the spatial layout of this tissue, but then also have individual cells that we can project to see, for example, what kind of clusters we have, similarity between these clusters, etc. So here's one view where you see this as an image. Here's another view where you would see individual molecules. So this was an in situ measurement. This is this OSM fish technology that was used, and you can see, so each little dot in here, it's not a cell, but is an individual transcript. So an individual molecule that was labeled with a marker, and then via microscopy was identified what transcript is actually at that position in that cell. So with certain assays, you can even look at the distribution of molecules within a cell, not only sort of looking across a whole individual cell, but you can even look at sort of distribution within a cell. That's possible too, and in some cases that's very insightful. And finally, another type of assay or visualization that is often used is when you have measurements that are not just transcriptome, but for example, the epigenome of individual cells. So this is data from an assay called ATAC-seq that measures essentially the accessibility of DNA for transcription factors and other machinery that's needed to regulate gene expression. And so you can, for example, in this case, you have a visualization of genomic profiles for clusters. So this is not per individual cell. Usually per individual cell, the epigenomics data is too noisy to really be insightful. So you can look at clusters and then look at measurements in there. And you see, for example, in this profile here, this green one, cluster number 13, has a very different profile. So there's some accessibility in the epigenome or in the genome here for the genome that is not present in the other clusters. So this could tell you something about which genes maybe are upregulated or turned on in these cell types that correspond to that cluster. And finally, of course, there are also many types of statistical plots. You'll see a lot of violent plots, histograms, bar charts, etc., in visualization of single cell omics data, but I don't think I'm telling you anything new here. Now, as I said, in terms of the visualization, there's not that much groundbreakingly new stuff that you need to consider, but what you do need to consider is how to bring this all together, right? So first of all, given that single cell and spatial biology is really still sort of in its infancy, you want to probably build tools that are not combining analytical approaches, or are not dependent on certain analytical approaches and tie that to your visualization. It's much better to separate these things because the analytical work is progressing very rapidly, as well as the visualization work. So if you can separate yourself from the analytical work, you'll be much more flexible in the future to support different types of analytical frameworks. And hopefully you'll be able to support the framework that everyone ends up using in a couple of years. What's really also critical is that you can build tools that combine these different types of visualizations in different ways, because sometimes you have comparative visualization, sometimes you want to bring in lots of different data types, and it really is highly dependent on the context. What I also strongly encourage is not to rely on a server if you can avoid it, because whenever you need a server and you want to make a visualization tool that other people should use, they need to set up a server, maintain a server. It's a lot of work for them. In many cases, they don't have the bandwidth and they'd rather not do that. So if you can make a visualization tool that doesn't need more than a web server, say, that's definitely a win. Building components that can be embedded with other applications, such as data portals, also very useful. And so the PubSub model for interactions, I mean, this is something that totally depends, of course, on what you want to build in terms of an application. This is an example from our work where we're using PubSub to communicate between different views. And so I just want to briefly point this out. So this is for a tool that I will introduce in slightly more detail in a moment, called Vitesse, that my lab has developed, where we're using a very flexible view coordination that allows us to tie together different data sources with different visualization types and then coordinate different aspects of these visualizations with each other. And this is, old school data visualization work. So the model that we use for the view coordination was described in 2003 in information visualization. So this is not something that we've invented or that you would need to invent, but it's critical that you build modular tools that can really support what I listed here again in terms of the an architecture that makes it easy for other people to embed your work into their tools and into their workflows in particular, because many people will use like Jupyter notebooks or R code to run the analyses. And if you want to help them with their work, that's where you would want to plug in. In terms of imaging data, I want to make a few quick comments here, and I realize I'm maybe a little short on time here. So I'll just point a few things out that I think are important to say about imaging data. So imaging data, as I said, lots of different assays that people generate for these kind of studies, and they can be optical, they can be mass spectrometry or mass cytometry based. You'll generally end up with a lot of different data formats. So what are you going to do about that? In particular, when your data is very high resolution, generally tens of thousands by tens of thousands of pixels, you often have high bit depth, meaning more than 8-bit, which is RGB. So you have often like 16 or 32 bits per channel. And then often you also have dozens of channels in some of these assays. So there are standardized image formats such as Omics, but one of the downsides of those formats so far was that either you had to convert these high resolution, high bit depth images into sort of like pre-generated tiles that you could then send to the client from a web server, or you need to run a specialized server that can basically generate image tiles on the fly using whatever parameters from mapping, from high bit depth to certain color mappings on the client. But so we developed a library that makes this all easier, where you basically we can point your web browser literally to an Omics TIFF file or a compatible ZAR file, which I'll tell you in a moment what ZAR is. And you can do all the rendering and all the data mapping in the client, and that really simplifies tool development for other people. So this library called Viv has become quite popular in the community, and a lot of people have started to play around with it. So ZAR, just want to point this out, is a very popular Python package to store chunked data for sort of retrieval at different resolutions and for different aspects of a data set. And that's highly recommended if you're working with large matrices, which is essentially large images. In terms of genome map data, I already pointed this out to you. So, you know, we developed a visualization tool called HighGlass back in the day for genomics data visualization. This is actually also an active project in my lab, but we integrated this HighGlass tool with scatter plots of different kinds and cell set managers. Cell sets could be cell types, but could also be other types of clusters that are being computed in this framework that we call VITES, that allows you to combine these different assay types in a very convenient way. And in fact, in terms of integration, one of the things that we have done in this HubMap project is we developed, I mean, my team is responsible to develop the user interfaces for this HubMap data portal. So you can browse through hundreds of data sets. And what is very convenient is that you can also visualize data sets directly in the data portal without the need to download any data sets, to install any software. So here's this VITES tool directly embedded in the portal. And you can see here's just an example for image data. So this is an assay called Codex, which measures protein markers across different channels. We have cell segmentations, and then we can map abundance of certain markers on top of these cell segmentations. So this is CD20 in this case, which is an antigen. You can see we have different clusterings. We have also different types of images. So this is standard pathology imaging, basically. Periodic acid shift stain. We have the scatter plots embedded here. You can play around with those directly in the data portal. You can make selections. You can save these selections, download the cells that you're interested in. And also, if you want to do more with that, you get a list of cell IDs that you can then feed into your analysis tools and continue from there. So that's what this, why I pointed out it's so important to build flexible tools that can be integrated with other frameworks. So here we really have React components that we can embed directly in a data portal, and allows us to show all sorts of data types in an environment that makes it really, really easy for biologists to access these visualizations, right? So that is a critical part of, I think, doing data visualization in an applied field like biology and medicine, is that you make tools that other people can actually use without having to bend over backwards or install software or anything like that. And I see Jan is already back in person here, so I'm going to try to wrap this up as quickly as possible. Another important point is, not only biologists will want to use your tools if you build good tools, other developers want to use your tools too. So integrating these tools not only into data portals, but also analytical ecosystems such as R and Python is critical. As I said, people use Jupyter notebooks, they use R, Shiny to make tools. And so what I think is critical is that you build tools that can be embedded in these environments and also support common data formats. So this VITES tool that I've mentioned before, it can be embedded in Jupyter notebooks. There's a Python package that allows you to generate these visualizations. Of course, it's all web-based, but you can write Python code to generate these visualizations because that allows you to tie them directly to analytical frameworks. It can be embedded in Shiny as well. It supports standard file formats such as this data format that is commonly used for single cell data and also for spatial single cell data these days. So here's a common assay called Visium by 10x that has a spatial component to it, and we can create visualizations on the fly with VITES. Vitesse can also, or will be able to, in a couple of days, do raycasting and three-dimensional tissue imaging. So you can look at volumetric data as well directly in this tool, where our imaging library can do that. And I just want to maybe point you to the website. So there's a new website for this tool. If you're interested in this project, you can check it out. You can play around with it. As I said, you can write some R code, Python code, whatever you want. There are plenty of examples as well. And I want to thank my many co-authors or my many team members who are working on this project in my lab, my collaborator, Peter Karchenko, who's contributing to this as well, our funders. And then finally, of course, want to point out, as I said in the beginning, I also have open positions in case you're looking for opportunities to work in this space. Thank you.
Okay, thank you very much, Nils. That was quite interesting talk, I would say. And it actually generated quite some questions. So let's maybe go quickly through them. So Laura Garrison is asking a question regarding the transition between the scales. She's wondering whether numerous cells, tissues, types are present in multiple organs, and how do you think about localizing these to specific organs or system sites, perhaps beyond the usual body maps that are just highlighting all regions?
Right. So I think that's an excellent question. And I think the body maps, in fact, are, I mean, they're useful for some things. They're useful for people who have never seen a human body and want to know where the kidney is or the heart. But I think they're not very helpful for understanding biology. In fact, they're making it much harder to navigate these spaces. So creating visualizations that map information that's more at the molecular level, which can indeed be similar across different organs together, and show, for example, tissues side by side that have similar cell type compositions or similar interactions between cells in a view that can be juxtaposed independently of where where the physical location of the tissue was. I think that's much more relevant to study data at this level. So I would say body maps are not particularly helpful. I think it's much more interesting to use maybe ontological annotations or the actual molecular data to organize these data sets and then navigate them using that way.
I hope that answered the questions. It was, at least for me, quite interesting to hear. So Alexander is then asking or wondering whether immersive visualizations such as VR or AR would be beneficial for the analysis of this data due to the spatiality.
I think, I mean, there is certainly, because the data is intrinsically spatial in this case, I think there is potential for VR or AR approaches. In the HubMap project, we have a team, not my team, but another team that has developed some VR-based tools to place tissue samples within the human body. So to help the people who extract the samples to place them basically within the body. I think that can be useful because the people who extract tissue from, say, an organ, they look at the three-dimensional object, and it's easier for them to place it back in a three-dimensional object if they can sort of see it in that view. You know, in many cases, the questions though that people are trying to answer are, I think, much more at a level of statistics and summarizations, and in that case, I'm not sure if you will always benefit from virtual reality or augmented reality solutions. I think one should always carefully weigh sort of the advantages against the disadvantages of those approaches. For example, accessibility is a huge issue, right? I mean, yes, you can argue that with my iPhone, I can do AR, but I'm not sure if it's the kind of AR that a scientist would want to use to analyze a data set that probably costs millions of dollars to generate. But you know, other people don't have that kind of infrastructure available to them, like headsets, HoloLens, whatever, and that limits the use of these tools to a very small number of people. And I think you know, figuring out what the tasks are and whether 3D representation is actually necessary, in particular AR, VR representations are necessary to answer those questions is I think a worthwhile endeavor before embarking on building a really cool thing that probably will get a lot of attention for it being cool, but if it's not helpful, I think that would be ill-advised.
Great. So there are many more questions, but I guess we will have time just for one now. So the last one is from Klaus, and he's wondering whether if you calculate the TSNE scatter plots in Vitaske, whether this is done, or if you do it within the browser, whether this is pre-computed or whether this is done live, because for big data set, this may take some time.
Generally, these are pre-computed. Yeah.
Yeah. So there are like another 3 or 4 questions, which we don't unfortunately have time to answer right now, but it would be great if Nils can stick around within the Discord channel, maybe take those later on. And
I will try to install Discord, and then maybe I can join you there. I have only a couple
I think you can also
left.
use the web version of the Discord without installation.
Okay,
But
I'll do my best.
I may be wrong there.
I'll give it a shot. Thank you. Thanks again, everyone, for the time and for the questions. And sorry for the next speakers for running a little bit over. Thank you.
No worries. It was really nice talk. Thank you very much, Nils. And in the last part for today, we will have a talk given by Thomas Bayer and Laliat Kumar. Thomas Bayer is full professor at the Medical University of Vienna, where he leads a quantitative imaging and medical physics team. He is co-developer of the first combined PET-CT systems worldwide, and he has a background in research and project management in academia and imaging industry. And lately, Thomas engages in the exploration of the human functional connectome using total body PET imaging. Laliat is a postdoctoral researcher in the Thomas groups. During his PhD studies, he's established a clinically viable computational framework to determine the cerebral metabolic rate of glucose in the fully automatic and non-invasive manner by using the synergetics information provided by the integrated PET and MRR. He is currently developing data analytics schemes for probing the human metabolite connectome using extended field of view coming from the PET-CT scan systems. So I'm really looking forward to their talk.
I think Thomas is not able to share his screen because he's not a panelist yet.
Oh, thank you. We will look into that.